Cells are the fundamental units of life, and their study reveals much about the functions and mechanisms of living organisms. Both onion peel cells and human cheek cells are often used in biological studies to understand cell structure and function due to their simplicity and accessibility. Despite being from different organisms, these cells share some common features but also exhibit significant differences. Onion peel cells, derived from the epidermal layer of an onion, are plant cells characterized by a rectangular shape and a rigid cell wall made of cellulose. This cell wall provides structural support and protection. Inside the cell wall is the plasma membrane that encloses the cytoplasm and other organelles. A prominent feature of onion cells is the central vacuole, a large, water-filled organelle that maintains cell turgor and storage of nutrients and waste products. The nucleus, which contains the cell's genetic material, is often pushed to the periphery due to the large vacuole. Other organelles such as chloroplasts are absent in onion peel cells since they are not involved in photosynthesis. On the other hand, human cheek cells are animal cells that do not have a cell wall. Instead, they are surrounded by a flexible plasma membrane. These cells are typically irregular in shape due to the lack of a rigid structure. Human cheek cells contain a nucleus that is centrally located, enclosing the DNA. Within the cytoplasm, various organelles such as mitochondria, which are responsible for energy production, and lysosomes, involved in waste breakdown and recycling, can be found. Unlike onion cells, cheek cells lack a large central vacuole and chloroplasts. Comparing both types of cells, one can observe the presence of a cell wall and a prominent vacuole in plant cells as key distinguishing features. In contrast, the absence of these structures and the presence of multiple small vacuoles and lysosomes are indicative of animal cells. These differences highlight the distinct ways in which plant and animal cells have evolved to fulfill their specific functions. Understanding these fundamental differences provides insight into the complexity and diversity of life forms.